Good morning, everyone. My name is Alan Lee. I'm a new assistant professor at the Department of Chemical Engineering and Material Science here at the University of Minnesota. My research falls at the interface of metabolic engineering, synthetic biology, and biocatalysis, as well as biomanufacturing. So I'm very excited to hear about all the leaders in the field, both from the North America and Japan. And I'm very excited, I'm absolutely thrilled to introduce Professor Hal Alper as our speaker. Professor Alper is a Cockrell Family Regents Chair in Engineering at McKenna Department of Chemical Engineering at the University of Texas at Austin. As a leading expert in metabolic engineering, synthetic biology, and protein engineering, he has been recognized by numerous prestigious awards and honors, including 2025 International Metabolic Engineering Award. Please join me in welcoming Professor Alper.
Well, thank you very much for that introduction and the invitation to be able to speak here today. So we do a lot of work in our lab that is really relevant to the types of things that are part of the symposium, but I really wanted to talk today and focus on the areas that we're working on in terms of enzymatic depolymerization of polymers. So just to either at least motivate, I wouldn't say motivate, maybe contextualize the challenge. Because 400 million tons a year of plastic is not really a motivating type of factor when we kind of see that. But this is the reality. So right this year, it's actually over 400 million tons will be produced this year alone. I'm an engineer and I like numbers, but I still don't understand what that number fully means and represents. So how much is 400 million tons? And it's actually 11 million tractor trailer trucks that are completely fully loaded. But that's not a number we can fully imagine since there's only less than 3 million in the United States. All right, that's 700,000 fully loaded double-decker A380 aircraft. Again, not a number that we can fully understand because there's only about 250 that have been manufactured in the world. So the thing that really kind of hits home is that 400 million tons is equal to the collective weight of every human on the planet. So we produce this much plastic this year alone, and that number is exponentially increasing in the amount that's being produced each and every year. So this really opens the door to needing to both find sustainable solutions for the end of life of plastic, but then also kind of illustrates just the scale and scope of this engineering challenge and problem that we have to face. So in the idea of trying to be able to establish this circular plastic economy, the concept is at some point in time, everything that we make will be some kind of post-consumer product will be collected. We can use some combination. It doesn't necessarily just have to be enzymatic processes. I'll talk about those mainly today, but through different types of processes to be able to break those down. In some cases, as I'll showcase with PET, that's a really simple process in respect to the fact that it has a ester bond linkage that we can break that apart back down to subsequent monomers. In other cases, we won't be generating original monomers, but we can still deal with that in a valorization type of standpoint to be able to, in some cases, repolymerize back and create this closed loop process. But like I said, in other cases, in a more generic fashion, we take this used plastic, somehow break that down. Either it goes into a monomer to be fully recaptured and goes to A type of circularity, or we have an off-ramp towards some sort of microbial or chemical upcycling and valorization. So the story starts with the idea of looking at PET, since this is a topic that a lot of people have been looking at, and there's activity that is abound for that. And really, this is a lesson of borrowing from biology. And kind of really fitting that story starts in Japan in that regard, in terms of an organism that was identified on a plastic surface. This organism, at least former name just recently changed, was Ideonella santiensis, was discovered in 2016 and published by a group in Japan of this organism that's able to colonize onto the low crystalline portions of PET plastic. And then it was found that there is an enzyme that is responsible for that breakdown of that ester bond from that PET down into the subsequent monomers of terephthalic acid and ethylene glycol. So this is kind of where we started the part of the process. This is a known enzyme. It had activity that was good for a microorganism in the environment, but pretty lousy for the activity that you would want for an industrial process. So our goal was to be able to greatly enhance the overall activity. So to do that, we took a machine learning or neural network-based approach to be able to look at protein design to be able to improve the overall activity of that enzyme. And so we used this model that was in collaboration with the Ellington group at the University of Texas Austin as well, this protocol called Mute Compute, which essentially analyzes all proteins in PDB and creates these microenvironments around each and every kind of amino acid, and then looks at that amino acid that's there and sees how well fit that amino acid is compared to the average or canonical enzyme. And the thought process behind that is the average enzyme that you, or protein rather, that you see within PDB is going to be a relatively stable type protein. And therefore, if we have mismatches, we can start to move that towards a point of more stability. And this has been shown in the past to be able to improve the thermostability of enzymes. So we kind of looked at that same type of approach to see if we can improve this PETase enzyme. And ultimately, that narrowed down to 4 major mutations that we found that can greatly enhance the overall activity. And we ended up finding and developing this as what we call fast PETase, because it's highly functional with respect to its overall capacity. It's highly active under a number of different conditions. It's stable with respect to thermostability, as well as to an extended pH range, although it's still not exactly where it needs to be for full industrial implementation. And it's highly tolerant to a variety of different conditions. So a lot of the things, and we were having the dinner last night, Romas was talking a lot about some of the work that they've been doing in terms of identifying mutations that are much more distal to the catalytic site. And we heard a little bit about that in some of the earlier talks as well. And here are similar types of things where some of the major mutations that were identified were not in that catalytic domain, but rather in the more distal parts of that protein. So therefore responsible for salt bridges as well as that kind of overall shape of morphology on there. That then led us to kind of showcase the fact that at this condition of 50 degrees C, when this was published at the time, outperformed pretty much everything that had been published to date in terms of the activity of the ability to take a piece of PET plastic and break that down to the subsequent monomers. We then found that these mutations weren't just specific to that one protein that we put in as the scaffold, but rather we wanted to evaluate this portability. And this is a kind of a fun protein diagram where all the various different petases are kind of superimposed on one another. And you see a very similar type of folding structure for these cutinase-based esterases. And there's a pretty conserved residue that we find. This kind of obviously the position number can change based off of the exact protein, but we found that this, trying to evaluate this portability of this identified mutation. And in particular, when we started moving this over to a variety of other scaffolds, whether it's CUT190, the leaf-cut cutinase, or a modified version of the leaf-cut cutinase, we were able to indeed enhance that activity through those mutations that were identified in that neural network. And then we expanded that scope very recently and found that across the board for various different other newer petases that had been discovered, we can increase that fold activity in a given operating range of temperature. So really the mutations that we can identify through this neural network approach tend to be relatively generic and portable, which allows us to be able to further enhance a suite of enzymes rather than just one singular one. Well, we kind of did the polymer analysis on there, so this fast PETase really just breaks down this plastic at the molecular level. We can look at that based off of different types of microscopy that you begin to see these pores or holes that form within the plastic until the entire piece is completely broken down. We then kind of went shopping to some extent. Pretty much everything's in PET, so it wasn't that hard to find various different packaging materials. And each and every piece that we had, we just take a hole punch of the PET, which was actually kind of ironic because the hole puncher itself was wrapped in PET, so we could kind of punch the hole of its own packaging. And of course, the thickness is going to be a little bit different, but in each and every one of those cases, we're able to completely consume that type of packaging PET and break that down to the monomers. We then started scaling up that process a little bit and literally take a handful of plastic, as you can see there, just kind of a cookie container that we just dunked into our beaker with our enzyme solution. This was operating at just 50 degrees Celsius. After a little bit of time, we can take it out and see that it becomes opaque in terms of the areas that are the thinnest in the regions. And then as time goes on, we eventually get to the point where there's nothing left to take out. And after about 40 hours, this thing is completely degraded into original monomers. We could see this visually on a much larger thing. So if we took this entire cookie container itself and dunked that into our enzyme solution, and over time, we can kind of see what happens in this time lapse. And the thinnest of the regions are those that kind of degrade the first. And then we start to see that opacity throughout the entire piece of plastic until eventually there's nothing left, and it's all a vat of just the terephthalic acid and ethylene glycol. So we can really showcase the power of having these enzymes this way. We also wanted to think about improving this rate, because there's practical side of degradation of plastic is the fact that it's dirty. It's not single stream. It's going to have other stuff on it. So we wanted to see what happens when we didn't have a nice pristine piece of plastic, and kind of looked at the impact of various different things that can foul that plastic surface, because at the end of the day, we have this enzyme that's trying to bind to this more hydrophobic surface and find this substrate that's actually a solid. And we find that there's a number of things that can actually decrease the overall effectiveness. And a lot of times it was in bacterial culture media, which is kind of interesting because we were studying the idea of being able to do recycling on these single-use plastics, especially in the biomanufacturing sector. And found that there is these key elements. In particular, it was proteins. It was other proteins, whether it was this case it was FBS or BSA, or even the protein itself, even the PETase function, when you have too much of it, binds to the surface of the plastic and actually prevents that activity from happening. So there's really kind of this interesting fouling that occurs. And you can kind of see this where we just kind of added GFP as that fouling agent. And you can see for PET plastic, as you begin to add more and more GFP, that piece of plastic becomes fluorescent as that GFP is absorbing to that surface. We found that there's a couple of different wash steps. Water works really well. Sodium hydroxide actually works a little bit better in terms of being able to wash that off and be able to enable that activity to then reoccur. It's not just a PET issue. This happens on polyethylene as well. Maybe if anything, polyethylene absorbs it a little bit more, but those wash steps can still ameliorate that, as well as even bio-based plastics. So things like PLA are also going to absorb that GFP and protein. So really, this kind of describes, one, a practical fouling aspect that can occur during the collection stage on plastic. But then second, the need to be able to think about this optimum protein content, because even the enzymes that we add could also foul that surface as well. And like I said, as we kind of do this washing step, we can begin to relieve that issue. So even plastic that had significant amounts of fouling from GFP, with our wash steps can actually remove that inhibition and allow for degradation. So that's challenge number one in terms of getting the difference between clean versus dirty plastic. The second challenge, as I sort of alluded to at the beginning, this organism, Ideonella, was isolated from specific spots on the water bottle, and it was actually the areas of low crystallinity. And you can see that the water bottle itself has a very different crystalline structure depending on where you are in that bottle, just on how it's made in that blow molding type process. And not surprisingly, the areas of low crystallinity, in terms of that top part, that finisher neck, and then the base on the center, which is kind of that little divot button that kind of sits on the bottom of the bottle, actually have low crystalline regions, and those are the ones that can be degraded most readily, whereas in turn, the areas that have high crystallinity have a really tough time with respect to the enzymatic degradation. We showed that we can actually do quick melting and quenching, and we can actually alleviate that where we end up melting these plastics into these hockey puck style components, which are actually quite thick. Just throw that right into our reaction, and we can actually degrade that completely. So really showing that that activity is indeed related to the crystallinity of that plastic. There's other ways that we think that we can get around the need to do this pre-processing, but at the moment, this is kind of where we need to be for getting that activity high. When we can actually get this to function, we can go through and take pieces of plastic. In this case, we took a piece of colored PET, so a green container, broke that down with our enzyme. We can recover the terephthalic acid by quickly crashing that out of solution. This mic done. Quickly crashing that out of solution with a low pH, and then recover that terephthalic acid and reconstitute that plastic. And you can kind of see the outline of that right here. With respect to this transparent film. So we're able to take what would have been challenges with mechanical recycling of multiple color pieces of PET and be able to reconvert that back into clear reconstituted plastic. So that kind of showcases a little bit of the effort that we've done with PET, but the plastic problem goes well beyond PET and beyond just the accessibility with respect to recycling. So PET kind of sits in this kind of bottom part here where it can be mechanically recycled a couple of times before the integrity is lost, but it's one of the things that we don't produce a lot of compared to some of the other type of plastics. Say these are some of the troublesome plastics where we don't have the ability to recycle those, or the sections up here where we have really high volumes of plastic, meaning that constitutes the higher fraction of that 400 million tons a year. So we started off in terms of looking at how we can deal with the next one, which is these polyolefins type molecules, the aliphatic ones, with respect to polypropylene and polyethylene as two challenges. So for that, we needed to go back to the beginning. So we didn't have that organism from Japan and that water bottle recycling facility to start off with things like PE. So we needed to go shopping on our own. So the aspect of bioprospecting. And we went to a number of different compost facilities and took samples across the various different waterways and into the compost piles. And not surprisingly, there was little plastic pieces in all of this. You can kind of see that in just the recovery of material in the compost piles, unfortunately, in the waterways. And that's the plastic we could see. And we know that there's lots of microplastics and nanoplastics that are around. So we wanted to see if this could be a really great place to find new organisms for activity. And then we developed this pipeline for enzyme discovery where we did environmental sample enrichments from this process. We then coupled that together with microfluidic and microdroplet-based selection processes, identifying microbes that then can grow on that plastic. Then did bioinformatic analysis to figure out what the key enzymes could be, identify and validate the targets, and then use machine learning to improve that overall activity. So I'll showcase a little bit of how that worked with respect to polyethylene. We took these populations and enriched them in various different types of polyethylene. And at the end of the day, our enriched populations were able to grow much better than those unenriched populations. And we were able to identify subsets of that population that grew better on different forms of polyethylene, whether it was linear low density, or high density. So that population kind of was with respect to the type of polyethylene plastic that we had in there. We then used microdroplets to further enrich and select individual microbes on that process. So going through and seeding these with some microplastic type particles in there to be able to look for a growth enrichment, and ultimately allowed us to isolate interesting organisms from that batch that we can then isolate down individually on a plate. That then led us to isolating in 2 different organisms that grow robustly on low density polyethylene. So here's just the standard square piece of that polyethylene. We identified one fungal host organism. This is kind of really easy to see in terms of its growth because it has a lot of melanin structure with respect to that fungi, so that can actually grow pretty robustly on there. This one is a bacterial host. You can kind of see the difference in terms of the coloration on there, where it kind of lays that kind of yellowish brown color on top of that PE plastic. But that's growing, both of those are growing on this PE as a sole carbon source. If we look at microscopy on there of the sterile control, it's a relatively flat surface. If we look at that, after we washed off the microbes, it's a much more kind of rigid type structure and mountains and valleys in terms of the act of degradation. We then did our differential RNA-seq to look at kind of pathway clusters and enzymes that we can identify. And ultimately, that allowed us to go from a sterile control to selected enzymes that we are able to showcase can begin to degrade that polyethylene as a pure enzymatic-based process. So this is outside of the cell and not having any of the cell activity in there. So that allowed us to go through and begin this process of degrading post-consumer polyethylene. We looked at 2 different ones, from a Goodfellow type film to one that was actually the plastic of a tea bag on there. And we were able to get overall weight losses, so looking at about 3 to 4% of that mass degraded in just a couple of days' time. So we're looking at rates that are, I think, initially pretty nice in terms of like 1 to 2% per day in terms of that mass loss and breaking down that polyethylene. We went through and found that there's a lot of stuff that's happening. It's a pretty complex reaction. FTIR shows that there's a lot of surface chemistry changes. The SEM shows that indeed there's a lot of morphology changes. The contact angle can showcase the fact that there's this massive change in hydrophobicity from this hydrophobic PE substrate to something that's been modified on there. And then LCMS shows a lot of different combinations of hydrolysis products at the end of the day. We then use the same type of algorithm of machine learning to be able to further improve this enzyme. And depending on the actual PE source, different mutants were able to get up to 100% improvement of that overall activity. So this is now beginning to reach 2 to 2.5% mass loss per day, which again is a pretty incredible rate considering this is something that would sit around for thousands of years if left to its own devices there. So that's kind of the beginning in terms of what we can do. We obviously said went bioinformatic and biomining and shopping for new organisms. And we found organisms that can also, different ones that can grow on PET. I showcased PE. But we have organisms that are beginning to grow on polyvinyl chloride, so PVC being that plastic that was on that bottom left of that original graph, are really challenging to grow off of and really challenging to even recycle type of plastic, off of beginning to have growth on PLA. And one that I'll talk with is even biodegradable compounds. So things like PHBs and PHAs and PLAs are kind of touted as a good replacement for petroleum-based plastics, but because they can be biodegraded. But that still means that they're in a linear type of utilization pattern. So we still spend the energy and water to make that, use it, and then we can compost it, but then we need to go back and make it again. So this linear type of fashion still uses a lot of CO2, still uses a lot of water, still uses a lot of energy resources. So if we can recover that carbon from that process, we can create this more closed loop type of standpoint. So to do that, we were able to, once again, take these compost materials, go through multiple passagings, find organisms of interest, eventually streak them out, find robust growth, and identify individual organisms that can be isolated, in this case for PHB degradation. And once again, the SEM imaging showcases the fact that an untreated PHB is relatively flat surface. The partially degraded kind of has these really interesting type of almost fractal-like looking spherulite crystals that are formed. And this organism that we identified can actually break down this PHB in the course of about a week. It can take a complete piece and just completely degrade it to nothing in terms of its monomers. So we started the process of trying to identify the putative depolymerases, mostly from sequencing that organism, cloning in some various different putative enzymes of interest, and then starting to analyze those. And this is off of homology modeling. We didn't do the RNA-seq analysis yet on this, but even just based off of annotations and homology modeling, we're able to find enzymes that, compared to the no enzyme control, can get about 4 or 5% mass loss in, this is about a week's worth of time. So we're still not at the same rate of the organism, but we're beginning to see enzymes of interest that can function there. So really the crux of this is that biology can enable the ability to break down these plastics and enable this circular type of economy. The first discovery of that one organism to break down the PET really opened up that possibility. And now we also, through various different ways of bioprospecting, have the potential to develop and discover those additional 1 microbes that can break down a wide variety of other types of plastics. So really the message is that biology can be an important solution for plastic waste in general. Machine learning approaches are quite powerful, I think, for the ability to engineer enzymes and improve their overall activity. A lot of times they're useful for finding some of these distal mutations that are away from that active site to allow for kind of this maybe potentially even portable type of mutation analysis. We showcased the fact that phosphatase itself can rapidly depolymerize PET plastics, and that these mutations were portable. Plastics can be reused in different ways. In some cases, we can use that for PET regeneration, since we are breaking that ester bond and forming that back again. In other cases, things like polyethylenes or other types of plastics, we might not be regenerating. For polyethylene, we're not going to generate ethylene, but we're going to be able to generate some sort of other oligomer that can be reused and valorized in a different way. So essentially, it's bringing that carbon and that energy back into the overall economy but it might not be going directly from polymer to polymer. And similar strategies with respect to kind of this enrichment process, metagenomics, and these various different pipelines can be used for a variety of other plastics. We showcase things like PE and PHB, but I also showcase those flasks where there's that potential for even things like PVC and other really challenging to break down and utilize plastics. So with that, I'd like to acknowledge, obviously, my research group. They're the ones who conduct the research that I was able to talk about today. The ones who are highlighted here in red are those who contributed to this actual set of projects in terms of the plastic work that we have going on in the lab. And once again, I'd like to thank everyone for both the invitation to speak as well as for your attention. I'd be very happy to answer any questions. So thank you.
We have time for several questions.
Thank you. Very impressive talk. And the case of the PET ACE and the plastic degrading enzymes and microbes remind me the case of the cellulose degradation systems. In the case of the cellulose cocktails, there is a enzyme called LPMO that can boost the degradation by the oxidation, in addition to the hydrolysis. So is there a possibility to use such kind of oxidase enzyme to boost the degradation of the plastics?
Yeah, thank you very much. And that's a great question as well on there. And there's a lot of parallels with the cellulose type of challenges. I think one of the first and foremost one is that crystallinity issue. Right? So low crystalline cellulose is easy to break down, high crystalline cellulose is not. The same type of thing also with respect to the PET. Very low crystalline is able to break down, very high crystalline is challenging. So these enzymes, and the enzymes that come from these cells are not just one enzyme in isolation, and certainly the combination with other enzymes, whether it is some of these oxidase type enzymes that can be used to start free radical reactions and start to break things down, or through this idea of even scaffolding like a cellulosome has, may be really important as we look forward to kind of not having to do all that pre-treatment. So I think certainly there's a lot of space open to draw from all of our knowledge and parallels of lignocellulosic material. So I think we should certainly be learning from that, not ignoring that, because they're both polymers to some extent.
Thank you very much.
Yeah.
Yes.
Thank you. Great talk. I really like the slide where you broke down the crystallinity across the bottle and how that impacted degradation. Is there any evidence that the microbes are producing small molecules like solvents to help liberate the plastic before they send the enzymes out?
So there's not as much study in terms of what those actual microbes are doing. The follow-up study that someone had done for that actually isolated microbe showed that it's only really colonizing in those sections as well. So I think there's definitely going to be some kind of creep that goes on as it goes through, but the thought process is that maybe some of it is like de-stringing and pulling that polymer chain out rather than actually doing anything to solubilize the actual surrounding environment. That said, for other types of plastics that don't have those ester bonds on there, so things that are much more hydrophobic in nature, like PE, it is found that these cells will start to form this kind of layer around the outside. So both with respect to chemical layers that they can deposit, as well as then the cells binding onto it. So really, that mimics much more of kind of a biofilm formation mechanism rather than a solubilization mechanism, but it's still kind of forming that chemical structure around it and then allowing the cell to, over time, be able to chew through it. So I think we're starting to see some of that, but there hasn't been, I think, enough study in terms of that natural, I call it natural, plastic in the environment, natural, but kind of that native, I guess, response to plastics in the environment.
Great talk. So my question is, for this degradation product, do you think there might be some toxicity issue, like for the intermediate? Because if you just use the pure enzyme, then you can measure the released monomer. But if you are using microorganisms, then do you see the intermediate or monomer can be degraded immediately, or there will be toxicity issue? And how would you quantify the plastic? For example, the plastic usually particles or sheet. So if the intermediate product can be degraded, so how would you model the kinetics? How do you model the substrate?
Yeah, no, it's a great question in terms of the kind of the logistics with respect to some of the experimentation, and that's partially why we move a lot to these free enzyme-based systems. So most of the data that I showed is off of pure enzymes, so the cell's not there, because these cells, if you're doing any type of enrichment, that means it inherently has some of the enzymes to break it down, but then it also has all the catabolic enzymes to bring that something, whether it's a small monomer or slightly larger oligomer chunk, into the cell, and then be able to catabolize that. And analyzing the downstream products from when there's a cell in there has been really challenging. We've done that a little bit, and it's hard to know when you see this thing that's formed afterwards, was it because of the enzymes it secreted, or was it because of an intermediate that went into the cell and they got spit back out? So it's hard to kind of parse out what's coming from that enzyme process versus what's coming from the cell-based process. So kind of something that would harken back to your area is certainly the idea of if you had labeled substrate of plastic, you can actually then watch the fate as you kind of go through on there. But I don't think people have really done that to date to be able to kind of map that out. So that's where we kind of stick to these enzyme-based processes where we can know that there's a direct one-to-one mapping of that reaction mechanism. With respect to kind of toxicity, as the rate at which these cells are growing is pretty slow compared to the rate at which we can enhance those overall enzymes. So if we can enhance that enzyme substantially, we're going to get a really high amount of terephthalic acid that gets formed, and that cell is not really adept to be able to culture on that high of a concentration of terephthalic acid, versus if it's kind of slowly breaking it down and consuming the terephthalic acid at the same time, it's not really hitting that tolerance point because it's kind of the two parts of the process are at parity with one another.
Thank you so much. Let's thank our speaker again.
Thank you, Ever.
Next, I'm